Hi, everyone, and thanks for taking the time to swing by our poster walkthrough, designing 3D printed braces for imaging the Caroline Anole, Anolis carolinensis, DewlapMusculature using DiceCT. My name is Jacob George, and my colleagues on this project were Dr. Ian Browne, Dr. Paul Gignac, Dr. Alex Claxton, and Dr. Haley O'Brien. So to start out, it's a little difficult looking at form function relationships, particularly trying to document them in small gracile structures or small gracile taxa. So for our study, our model system were anoles of the genus Anolis, which are small lizards that are present in North, Central, and South America. So these guys they're kind of a common study taxa. They're normally revolve around their behavior, though, or ecomorphs, and they're very diverse. They take up a lot of ecological space throughout their range, but they're all very small. A lot of them are generally about this big. But an interesting thing about them is they have these specialized dewlaps, which they use for internet-specific communication. So you can see we have an image of a CT specimen on the left with the second serrata brachials highlighted in blue. And in the other image we have here is actually an anole with the dewlap popped out. So they use these, they flash them around to get the attention of both members of their species as well as to ward off predators. So here, this is the actual full structure of their hyoid bones. So it sits like this along their throat. And we have our lingual process, our main hyoid body, and our hypohyals, our serratohyals, epihyals are down here at the tip, your first serratobrachials, your epibrachials, and your second serratobrachials, which are the big boys because they are the ones that swing out this way to drive that dewlap action. So obviously, as you noted, we had a bit of a problem. So going back, there are actually two competing hypotheses on the mechanism of the extinction of the dewlap. Both of them are pretty uniform in the idea that the second serratobrachials are swinging out. So these bones here swing out this way, and you open the dewlap fold. Where they conflict, though, is around the other bones. So these two studies are Bells and Font and Rome. Bells posits that the hyoid apparatus is kind of shifting forward and down, so this way towards the snout, and this leads to and then with this action, the second serratobrachial swing out. Font and Rome, however, found that the hyoid body, they concluded that they were being pulled back, so posterior towards the back of the jaw, and then the second serratobrachial swing out. So we have one idea that's kind of this consistent motion down forward, and the other that is a bit of a displacement. So in trying to answer this question, it was a little difficult because anoles, as noted, are very small. And what we ran into, another issue, was that the specimens we were able to acquire were already fixed upon acquisition. So one thing we thought to try was maybe try to use some DiceCT to see if we could answer questions about how this mechanism is actually working, especially since no one has taken a look at this conflict in 30 years. So our goal with this was to design an apparatus that could achieve this feat of being able to analyze the anole dewlap musculature in an extended position in the most efficient and cost-effective way possible. So we went through a bunch of prototypes. So a couple of things, our structure had to be low density, it had to be stable, and it had to be small and intricate to be able to interact, to be able to fit with the small size of our specimens. So we went through a couple initially trying store-bought clips, such as alligator clips, wall clips, etc. But these all proved to be too large to work with our very small anole specimens, which again are like that big. So we went through a couple of 3D print designs. Since we have 3D printing facilities on campus, we figured we would maybe try to make our own device to try to do it. So we went through a couple different designs. Initially, a lot of them were wrapped around the idea of popping the dewlap open with the fold and then pinning that fold open, therefore keeping the second serratobrachials in a fixed position, in an open position. However, this proved to be too difficult because even with attempts to rehydrate the specimens, it was just too too much of the elasticity of the dewlap fold had been lost. So this dewlap fold could not actually open, and it just hindered our ability to manipulate these second stratobrachials. So that's when we moved in a completely different direction for our final design, which was actually a 3D printed brace harness with filament. So the idea is kind of like how you see with like deep sea fishermen, where they put a harness around their waist, and then you have an insertion point where you put the rod into, so you don't throw out your back when you're trying to reel in a massive marlin. So we added little tabs and teeth, little tabs that could fold up, and teeth that could grab into the skin of the specimen to hold it a little more securely. We ended up having to remove the tissue fold due to this loss of elasticity, negate our ability to manipulate the hyoid, the second stratobrachial bones, and we secured it with surgical suture since we had to transport it for scanning at the micro facility at University of Arkansas. This helped give extra security and make sure the specimen didn't move or that this brace didn't move. So, an idea of the scale we're looking at. This is the actual size. As you can see here, this is very small. These little details. There's the filament. There's actually a little brace, and you can see the little tabs are popped up. And this is what we put down the waist of the specimen. So our lessons learned and recommendations after this are that the Anole dewlap is too small and delicate for manipulation by readily available items, particularly in small species such as carolinensis. Future work could be attempted on larger species such as Anolis equestris, which is decently bigger, but we were unable to acquire for this study. A lifelike position is no longer feasible for our dewlaps while fixed unless the dewlap tissue is removed. So too much of that elasticity is lost. It's just an elastic fold that can be pushed out. So once the elasticity is gone, it cannot be opened anymore by regular means. So it has to be removed. So, and we also greatly support having 3D printing facilities in labs, and particularly biological labs, because they give you the ability to analyze unique structures and morphologies using novel methods and designs, so you don't have limitations of what can be acquired already on the market or ideas people have already done. So here are our references, as well as our acknowledgments, which are the York Micro Facility, Manon Wilson, Lyndon Colvin, OSU CHS, and the National Science Foundation.